a little material left to cover. We're mostly not going to cover chapter 9 before the exam. There are a few selected slides I want to go over, and that's both so we keep on track with the next unit, because the reason we're over is because we were a little slow on unit 1, so we don't want to be constantly a day late. And the other reason is that there's some very basic information that I think is really suited to flashcards and therefore hopefully easy questions on the exam. And I think they should be points that should be easy for you to earn on the exam, so I want to give you that opportunity. Because a lot of this stuff with the action potentials and the synapses involve a little more critical thinking, meaning a little more deep reading of the questions, so they can be a little bit harder. Okay, general questions before we finish up our content?
Hey, so for the anatomy lab, do you know if it's going to be changed at all the way it's taught? Because right now it doesn't really have to do with lecture. And it's, yeah, I don't know.
So there aren't a ton of links between anatomy and lecture, but they're going to get closer together as we go forward. Part of the reason for that is in lecture we needed to kind of set up the basic chemistry and basic understanding of cells before we could get started on actual systems. So there are no good ways really to link that to the anatomy lab. And the other thing is that we don't really have a unit on bones, but bones are crucial for anatomy, so we tried to cover those first. As we move forward, our next unit in lecture is going to be on muscles, and we'll be doing muscles at the same time in the anatomy lab and in physiology, so at that point they should sort of integrate together a little better. Yeah, and that's basically the answer. Honestly, the lecture portion of this course, as an anatomist, I personally feel that this course is mostly a physiology course. I don't know if a physiologist like Dr. Schlater would agree with that statement, but that's just kind of how it is, unfortunately.
And then one other question I have for lab is, I have no clue if you come up with the quizzes or not, but why do we study on the slides, like kind of like kid images, you know, like not real stuff, and then on the quizzes it's like a actual picture of a brain?
Luis has been setting up the quizzes, so I actually was not aware of that until you guys brought it to my attention, so thank you for that. Part of that is because in the in-person lab, the way quizzes work when this course is in person is that all those quizzes take place on the real materials in the first 10 minutes of lab. So they're just tags on physical images or on like actual bone or actual muscle. So I think he was trying to replicate that, but I understand that that's not super fair for people who are taking this course all online. Although I will say the scores between people taking this online and people taking this in person, despite the fact that there were pictures of actual cadavers, everybody did the same. The scores were the same. We checked after you guys brought it up to me. I talked to him about that. I'm going to make sure that for the online stuff moving forward, there are lots of images of the actual cadavers, so that's a little more equivalent moving forward. But that's why it's set up like that.
Yeah, it just seems like my lab's the hardest part of my entire course schedule, which is just kind of goofy. But
yeah, I agree. Are you taking this all online or are you in person?
Yeah, I'm taking it all online, so that's probably a part of it. But
yeah, that's probably part of it. So the TAs hold 3 hours of review hours every day except for Saturday. So to work on that and kind of emphasize that a little more, I would recommend signing up for those. I don't know exactly what they're doing in terms of virtual review hours, but I do know that people don't tend to go to review hours, so I'm sure they'll be happy to help. You can also always sign up for appointments with me for that stuff. Like, I'm an anatomist and I would frankly prefer to be talking about anatomy all the time. So I'm happy to help you out with that. Unfortunately, at this point, I can't really change the balance of topics in the course, and I'm not the only person who teaches it, so it's not really up to me.
All right, sounds good.
Okay. So that said, let's finish up the synapses and do some very quick stuff on the general setup of the nervous system. Okay. So when we left off, we were talking about postsynaptic potentials, which are just what's happening in the sort of second cell in our pathway after we release a neurotransmitter from our presynaptic cell. And so most of this unit, we've been focusing on that presynaptic cell, the building of the action potential, propagating it down the axon. Now we're in the postsynaptic cell. So when we left off, we had gone through a flow of just how this works in the postsynaptic cell, which is that when we have many action potentials coming in to our postsynaptic cell, this is going to release more and more neurotransmitter. And that's because if we have a lot of action potentials, that neurotransmitter is still kind of hanging around, and the stimulus effect in that postsynaptic cell. We talked about how graded potentials degrade over time and over space. If you're firing lots of neurotransmitter over and over again, it's like you keep buzzing it with a stimulus, so it doesn't have total time to dissipate. So this means we end up with more channels that are being either opened or closed, depending on whether the neurotransmitter is excitatory or inhibitory, and depending on which ion it's acting on. But the general thing that's happening is that the neurotransmitter is doing something to the channels, affecting membrane permeability to individual ions, which means that whichever ion it's either letting more of in or less of in or out, is then able to move less or more, which affects the hyperpolarization or depolarization of that postsynaptic cell. So that we had talked about, we'd kind of gone through the inhibitory version. This pathway happens to be ending in hyperpolarization, if we ran through an example. Excitatory synapses work the same way as inhibitory synapses. It's just that end result that's different. So at the end of an excitatory synapse, we're going to end up with depolarization of our postsynaptic cell, which means that we might be thinking about different neurotransmitters, and really we're going to be thinking about different primary ions that are going to be responsible for depolarization, and therefore we're going to be thinking about different ion channels. So specifically, we should associate in our head excitatory synapses with sodium and calcium channels. And that's because the electrochemical gradient for both sodium and calcium is into the cell. They're both positive ions. So when we open channels in an excitatory synapse for sodium and calcium, we have positive charges flow in, which depolarizes the cell. And remember that depolarizes means we're bringing that cell closer to zero, the membrane potential. The other thing that could happen, so there are lots of ways you could get to a number closer to zero. The sort of second most common way, if we're not looking at the sodium and calcium channels, with sodium and calcium channels opening and sodium and calcium coming in, is that if we close potassium channels, remember potassium's electrochemical gradient is out. It's a positive ion, so if it flows out of our cell, the cell becomes more negative, becomes hyperpolarized. So if we were to close a potassium channel, that would mean that we don't have this leakage of charges out, which would also have the end result of making that membrane potential of the cell a little more positive, depolarizing the cell. Okay, so those are three ways we could get to an excitatory postsynaptic potential in an excitatory synapse. So we could open a sodium channel, sodium moving in, which would create an excitatory postsynaptic potential. And if our potassium channels close, the potassium can't move out, we'd also get an excitatory postsynaptic potential because we're not losing charge. So any sodium that comes in kind of gets more bang for its buck. Okay, yeah, we'll come back to that at the end if we have time. Okay, this is just a reminder, you've seen this slide before, but this is just a summary of all the different types of ion channels we've talked about and their locations in the cell. So this is another memorizable thing potentially for the exam. So we remember we have our leak channels, which are basically everywhere because their job is to create the resting membrane potential of -70. So when we talked about the membrane potential, we talked a lot about sodium and potassium and how they're moving and how we end up with this balance between the sodium equilibrium potential and the potassium equilibrium potential, where we're closer to potassium, whose equilibrium potential is very negative because the membrane is more permeable to potassium. So that's happening through these leak channels. They're always open. We don't really talk about opening them and shutting them because they're basically creating our baseline. We talked about our ligand-gated channels kind of in two ways. The first time we thought about them a little bit was when we were talking about stimuli coming in and creating our graded potentials in our neuron. And we now know that that's basically what we're talking about, again, as we talk about neurotransmitters. When the neurotransmitter gets released from our presynaptic cell and binds on to either our ionotropic or metabotropic receptors in our postsynaptic cell, that's an example really of a ligand-gated channel. And these are going to be found in parts of the cell that receive signals. So this is mostly in the dendrites and in the cell body. Our voltage-gated channels, we wanted to associate in our minds with the action potential. So depolarization, largely due to sodium coming in, and then that hyperpolarization, that after hyperpolarization, and then sort of stabilization after the action potential as potassium leaves the cell and then comes back. Okay, so those are going to be found in the axon, especially in the axon hillock. And then finally, we had calcium channels. The only place we really think about calcium channels is in the axon terminal. They're responsible for releasing neurotransmitter. So when calcium comes into the axon terminal, that's the trigger to release those synaptic vesicles, which are our bags of neurotransmitter. Okay, so the final thing we want to know about synapses is something called neural integration. And we have a couple of ways we think about neural integration. We're going to think about something called summation, something called frequency coding, and we're going to think about something called divergence, convergence. Really, we're thinking about things that are going on at the axon hillock, which is kind of our integration center for the whole neuron, because that's what leads into the axon. So what we're really trying to depolarize to send off an action potential is the axon hillock. Okay. So an action potential gets triggered if the membrane threshold at the axon hillock gets to -55, right? -55 is our threshold. So if we get the axon hillock to -55, we fire off an action potential. So we've talked about this before, that if that potential is below threshold at the axon hillock, no action potential occurs. And we also talked about how even if we're above the threshold, even if we have a lot of excitatory stimuli, we're going to fire off an action potential that's exactly like every other action potential. we had a name for this principle.
Isn't it the all or none?
Yep, this is the all or none principle of action potentials. Okay. When we see these words divergence and convergence, when we're talking about neural integration, really we're kind of talking about the shape of the neuron. All we're saying here is that when we're thinking about divergence, we've been tracking through one neuron that basically just tracked down the axon to like one axon terminal. When I draw axon terminals, I usually draw two, which you may or may not have picked up on. Divergence is just this concept that says that while we do have one axon, we'll often have a split at the end of the axon into many axon terminals. So one presynaptic neuron can signal to many postsynaptic neurons. So there would be a synapse here, a synapse here, a synapse here, a synapse here. And so we call this divergence because basically that signal from our presynaptic neuron is diverging and sending reactions into many different pathways, basically. Convergence refers to the fact that if we're looking at a postsynaptic neuron, we've been looking thus far at just one synapse, but a postsynaptic neuron is actually going to be receiving input from many different neurons potentially. So as we see here, all these sort of red and green lines, anytime your book uses red and green, it's using, I think, green for excitatory, red for inhibitory. So if we're looking at a postsynaptic neuron, whether it's going to fire an action potential or not, usually has to do with many different signals it's receiving that it sums together, as opposed to just one signal from one neuron. So it's a little more complex. So this is convergence. So all these signals will be different stimuli, which would then have excitatory and inhibitory effects. And then we'd have our integrator, the axon hillock, where we actually kind of decide whether we're going to fire off an action potential or not. Okay. So convergence is linked with summation. We've actually really talked about summation before, just using slightly different terms. So when we talked about graded potentials, we talked about the fact that when we have stimuli coming in, they can add up in time. So if we have one stimulus and it keeps firing off, but we don't have time for that graded potential to degrade, they're going to add together. So that would be an example of summation. We also talked about summation in terms of space, so how they sum graded potentials sum spatially. So if we have one here, they're both coming together. If we measure at a third point, we'll have a little bit of the effect of each of those. So that would also be summation. So we're just kind of pulling back this concept, and now we're adding on our knowledge of synapses. So now we know that summation is really going to be happening in the postsynaptic neuron. And it's going to be determined by these inhibitory and excitatory postsynaptic potentials that we're creating. So we're going to have neurotransmitters coming in. And those are really our stimuli here. And we're going to sum them all together, both spatially and temporally, at the axon hillock, our integrator, and determine whether we're hitting threshold, in which case we fire off an action potential. If not, then we don't. Okay. So this is just a return to that topic from before, but now kind of layering it onto our knowledge of synapses. Okay. So frequency coding it is kind of linked to this idea of temporal summation. So if we're looking at the axon hillock and we're trying to figure out whether it reaches threshold, if we have more frequent action potentials, if we have, therefore, more like stimuli kind of in a row on that postsynaptic neuron, they're going to sum together, depolarize the cell to threshold or not. But if we have a lot of excitatory action potentials coming in, right, depolarization in the presynaptic cell, releasing an excitatory neurotransmitter, creating an excitatory postsynaptic potential, we'd end up with more depolarization. And as it hangs around, possibly more action potentials firing off from that second neuron as well. So if we have more action potentials coming in through this frequency coding in that presynaptic neuron, another way we can kind of think about this is that if we have depolarization coming to the axon terminal over and over again in kind of steady pulses, if we're doing that quickly, we're going to end up with kind of more and more calcium hanging around in the axon terminal. We talked about how that depolarization from the action potential is opening calcium channels in the axon terminal, which is what triggers the release of our synaptic vesicles and therefore our neurotransmitters. So if we have more action potentials in our presynaptic neuron, that charge hangs out in our axon terminal longer, we get more calcium channels open. More calcium means more neurotransmitter released, which means we would have more of an inhibitory or excitatory postsynaptic potential in the next cell in the chain after that synapse, because we have more neurotransmitter and every kind of individual particle of neurotransmitter is binding to a receptor and having an effect. So if we have more neurotransmitter, we have more an effect. Okay. So this is the sort of chunky topic I really wanted us to get to for today. Something called presynaptic modulation. So modulation is kind of an adjustment, and presynaptic should tell us that this is happening before the synapse. So we're thinking here about the presynaptic cell. So the one firing off our original action potential, the one that's going to release the neurotransmitter. So when we say presynaptic modulation exists, that means that we can actually have another neuron kind of come in and affect how much neurotransmitter that cell is going to release. So we can affect the amount of neurotransmitter both by the frequency of action potentials firing down that axon in our presynaptic cell, adjusting the amount of calcium, but we can also have a secondary kind of modulator neuron come in almost directly to the axon terminal, also adjust the amount of calcium, and therefore adjust the amount of neurotransmitter that's going to be released into the synapse. Specifically, remember, we're thinking about that space between the two neurons, so into the synaptic cleft. Okay. So we're going to be thinking here about axoaxonic synapses specifically. When I say we're thinking about axoaxonic synapses, I'm saying that the axoaxonic synapse is going to be between the presynaptic neuron of kind of one pathway and then this sort of modulator neuron that's coming in to affect the eventual release of neurotransmitter. So we're going to have a kind of second synapse that we're thinking about here when we're trying to figure out whether our second cell in this kind of chain is going to fire off an action potential. Okay. So since we're thinking about modulation here, modulation means we're changing, we're adjusting. So we're going to have adjustments in both directions. We're going to have presynaptic modulation that increases the effects of that neurotransmitter, increase the release of a neurotransmitter. We're also going to have presynaptic inhibition, which kind of decrease the effects. So here we see this structure. So the real pathway that we're tracking this presynaptic neuron to postsynaptic neuron. And our modulating neuron is coming in here from the side. It's either going to facilitate the release of neurotransmitter here, or it's going to inhibit the release of neurotransmitter here. Okay, so even though this is an axoaxonic synapse, we don't think of this modulating neuron really as like a presynaptic cell. If we were to talk about them that way, we'd have to kind of rename everything in this pathway. So it's easiest to think of it as like presynaptic neuron, postsynaptic neuron here on the main pathway that's working normally, axon terminal to dendrites or cell body, and then as having this sort of modulating neuron off to the side. So the difference between facilitation and inhibition is kind of like our differences between excitatory and inhibitory stimuli, postsynaptic potentials, etc. We're kind of turning on or turning off stuff. So we're going to start here on this left graph with presynaptic facilitation. So in presynaptic facilitation, what we're going to see is, let's look at this neuron first so that we know what's going on. So here we have neuron X. Neuron X is receiving signals from several different presynaptic neurons. So we'd say that axon X, right? This is an example of convergence. We have a convergence of signals, a convergence of synapses coming here to neuron X. So we can see that the neurons coming in are neuron D, neuron C. We also see neuron E here, but it's important to look closely because neuron E here is not a direct synapse with neuron X. What we see here is an example of this modulating neuron, this presynaptic modulation. You can see that neuron E is actually forming a synapse onto neuron C, right? So this is kind of an axoaxonic connection here. So E is not directly affecting X. What E is doing is affecting how much neurotransmitter C can or can't release. So if we look at a graph of the membrane potential over time, like I said, green means we're thinking about something excitatory. That's just how your book displays them, kind of helpfully. Green means go, right? So we can see here what happens in our postsynaptic neuron. We're putting a voltmeter at the axon hillock. Our postsynaptic neuron as each of these presynaptic neurons fire off. So we can look at C, right? Let's first look at C. It's green. It's excitatory, right? So it's telling us that this would create an excitatory postsynaptic potential. So we can see, bloop, up, C. If it fires alone, that's why it's just a pink arrow above C. If it fires alone, it gives us a little bit of excitation here. And we go back down to rest because it didn't hit threshold at the axon hillock. Same thing's happening with D. So D, also green, also excitatory. So D fires off, excitatory postsynaptic potential, also doesn't hit threshold in this specific case. So we go back down to rest, but we see that little bloop up closer to 0. And we see if E fires off, so an action potential comes down E, that depolarization, and E releases its neurotransmitter, it's not synapsing onto it's not synapsing onto neuron X, it's synapsing onto the axon, basically, of neuron C. So if E fires off, I've kind of answered this question at the bottom, but why are we not seeing a change to the membrane potential here if E is firing?
Because C has to fire too to actually impact the dendrite.
Yeah, in order for a neuron to directly affect the postsynaptic neuron, it has to actually have a synapse on that neuron. So we'd say here that a modulating neuron, right? If a modulating neuron fires and it's just the modulating neuron firing, we're going to see no effect in our postsynaptic cell. Where our modulating neuron really does come into play here is this C plus E. So C plus E tells us that both C and E are firing. And whoop, we can see that that hit threshold, and then we have our action potential firing off. So why does C plus E create an action potential when neither C nor E did alone?
Because the modulating neuron attached itself to C and it let more neurotransmitters out to
Yeah,
hit threshold.
yeah, exactly. So E here, this modulating neuron, it's excitatory, but what it's doing is like, it's doing facilitation, right? Presynaptic facilitation. So it's facilitating more release from C. So if they fire together, when E fires off, it allows C to release more neurotransmitter, which means we end up with more excitation in the axon hillock, because more neurotransmitter means higher postsynaptic potential, higher excitatory postsynaptic potential, which means we manage to hit threshold. So we see the whole axon potential. Why do D and E not have that same effect? So D and E in this diagram.
Because it's not attached to D.
Exactly, yeah. E doesn't have an effect on D. It's not modulating D, it's modulating C. So that's presynaptic facilitation. Presynaptic inhibition is just the opposite. So here they've renamed the neurons. So here now our postsynaptic neuron is neuron Y. So postsynaptic neuron is Y. We still have our voltmeter at the axon hillock. So now we have presynaptic neurons G and F plus modulating neuron H, we can see is modulating F. So if we have F fire alone, F, it's green, it's excitatory. So we can see that we have an excitatory postsynaptic potential that doesn't hit threshold with F. Same thing for G, excitatory. So we have a slight depolarization if G fires off alone. If H fires off alone, nothing happens, right? H isn't synapsing on Y. This is the cool part. If F and H fire at the same time, so H is modulating F. So here we can see the effects of H on F. This is our example of presynaptic inhibition. So when H modulates F, F can't release as much neurotransmitter, which means that we have no effect on the membrane potential in this case. It barely released any neurotransmitter at all. It's not having an effect on the axon hillock as that signal degrades. So that's what a modulator neuron that does presynaptic inhibition would do. Basically turns off the neuron that it's attached to if it fires. Here they have showed us an action potential firing off, but if we look, it's because F and G have fired together. So F and G are both excitatory. If H isn't firing, they're going to sum together. This would be a spatial summation, right? So they're going to fire together. Each of them gets us a little bit towards threshold until we hit it, action potential. And then finally, we have if all three fire together, we can look back, we see that G on its own isn't enough to hit threshold. So if F, G, and H fire together, because H turns off F, it's like we only had G fire off. So we see that same just small depolarization that fades, like when we just had G. Okay. Questions here about modulation? So this is just kind of a conceptual framework. So do expect to see an example of something like this on your exam. So as you're studying, kind of practice for yourself what modulating neurons on different types of synapses would do. When you see a question like this on the exam, just make sure to go slowly, because it doesn't have to be a difficult concept, but it's one you'll have to think through both the pathways, think through that original presynaptic to postsynaptic pathway, and also the effect of the modulator. Okay, so that was an example of an axoaxonic synapse, exciting or inhibiting, so facilitating or inhibiting, here if we're thinking about that, a synapse. This will be a selective sort of connection. It's going to have a selective effect because it really is just affecting the release of neurotransmitter here on the neuron it's synapsing with in that axoaxonic connection. So even if this neuron C, right, if we had divergence, neuron C sends out signals to lots and lots of different neurons. E, this axoaxonic connection of E and C, would be specific to this specific axon terminal. So if C had another axon terminal off the page somewhere, E wouldn't really affect that because it's synapsing here directly at the end of the axon terminal. So that's why it says this is selective. Our axodendritic and axosomatic connections, which are basically all the other synapses we've looked at, they excite or inhibit the entire postsynaptic neuron, right? So they're where we're talking about summation in that axon hillock. So we think of them as non-selective, because then if that axon in that cell branches out and synapses with 100 other neurons, all of them are going to have that effect, because that effect from the axodendritic or axosomatic connection, because they were exciting or inhibiting the postsynaptic neuron as a whole, which means that what they were doing, their effect is really just, are we firing off an action potential or not? Okay. So this is just a summary of the neurotransmitter action. So how we release neurotransmitter and kind of what it does. So here we see our presynaptic neuron, our presynaptic neuron here in brown, and then our postsynaptic neuron. So in our presynaptic neuron, what would happen? So our action potential would be conducted by our axon, opening up those voltage-gated calcium channels, releasing a neurotransmitter. In this case, it's telling us that it's excitatory. Okay. We can see that these are axosomatic connections. So they're connecting to the soma of the neuron, the cell body here. What that does, what that neurotransmitter does, is it opens gated channels, specifically ligand-gated channels. We think of this as a chemical signal, right? We talked about chemical and electrical signals. We saw that the electrical ones were really just gap junctions between cells. So we don't think of them as this sort of type of synapse that we've been tracking through this unit. So these are chemically ligand-gated channels, which means we're going to have some effect on the movement of ions. So one effect might be that sodium would diffuse in. Sodium, positive ion, it moves into the cell, causing depolarization, which we could also call an excitatory postsynaptic potential. So that would have an effect here at the actual synapse, right in this local area of where the synapse happens. As we move farther away from that actual synapse, we'd have less and less effect, but we'd have some potentially in the axon hillock, depending how far it is, depending how much neurotransmitter was released, depending how big that depolarization was. So the axon hillock will have some effect if that axon hillock depolarizes to threshold, -55. Then we're going to fire off an action potential in this postsynaptic neuron, specifically by first opening our voltage-gated sodium channels, and then turning off that signal by opening the potassium channels to repolarize us after hyperpolar, and then have that little after hyperpolarization. So we'd have an action potential fire down our second axon. So this is just a full summary of what could happen here. Okay. So a little tiny bit about the central nervous system now. Okay. This is a lot of words, but we're going to boil this down into like 2 facts, I think. So the 2 facts we want to know here about this diagram is that one of our divisions when we're thinking about the nervous system as a whole is that we have a central nervous system for information processing, really kind of acting as an integrator if we think about what we talked about in homeostasis. We have a peripheral nervous system where we're receiving signals from other parts of the body, we're sending out signals to the body. It's happening in the periphery. So our central nervous system is just the brain and the spinal cord, both of which contain lots of neurons. And our peripheral nervous system is everything else. So nerves in your arms, in your legs, in your abdomen, peripheral nervous system. So that's one way we divide the nervous system. And in our next unit, we're going to be talking about this second division between the somatic and autonomic nervous system, but we don't need to know that now. Just want you to know central nervous system, brain and spinal cord, peripheral nervous system, everything else. Okay. Second fact from the slide we want to know is that when we're talking about the nervous system, we've been focused thus far entirely on neurons, but we also do have supportive cells called glial cells. So neuroglia exist, they count as part of the nervous system, and they support the neurons, but they don't fire off action potentials themselves. So they're not excitable. We don't think about them depolarizing. They're just there kind of covering the neuron, doing various jobs that protect the nervous system. Okay. So our 2 facts. CNS versus PNS, and then glia exist. Cool. Second thing we want to know is that we can break down neurons in one other way. So when we were thinking on that previous slide, we had central nervous system, brain and spinal cord, peripheral nervous system, literally everything else. When we're thinking about a neuron, we can say whether it's in the CNS or the PNS, the central nervous system or the peripheral nervous system. We can also talk about what it does. We can talk a little bit about the signal. So we have nerves that are sensory. So sensory neurons sense something out in the body. We have motor neurons. Motor neurons send signals out to the body, so potentially to, for example, muscles, but we'll also see motor neurons going to glands. And our final thing is that we can have association or interneurons. They're going to be in the CNS, and they're basically kind of connectors between other neurons. We wouldn't think of them as sensing something or sending out a signal. They're kind of moving signals around within the brain or the spinal cord. So this is just glial cells exist. There are actually lots of them. Most of our nervous system is not neurons themselves, but these protecting cells. And there are 5 types. So we want to just recognize that there are 5 types, and that'll be it. This is where we're going to stop. We don't need to know big details. We're going to do just word association here. So astrocytes, we're going to say are associated with something called the blood-brain barrier. All the blood-brain barrier means is that your brain, your nervous tissue is protected from a lot of things that are in your bloodstream. A lot of things in the bloodstream could be damaging to your brain. So there's this sort of protective layer between what's in your blood and what's in your brain itself. And this is one of the big jobs of astrocytes. Ependymal cells are, I would not consider as important. They line cavities, specifically the ventricles that we've talked about in anatomy. So we looked at the brain and the skull in anatomy. They'd be lining those areas that contain cerebrospinal fluid. Microglia are phagocytes, which basically means they're kind of like your immune system for the brain. Phagocytes are cells that eat things, basically, so they eat them and break them down. So microglia are going to be protecting your brain from various types of infections, potentially. And then we have two different types of cells that form myelin. So we talked about the myelin sheath that can make our action potentials go faster down an axon because we're jumping between nodes. We don't have to fire off the whole membrane. So those would be your oligodendrocytes and Schwann cells that make that myelin. Okay. So that's it.